Today, my guest is Xuan Lu from the Max Planck Institute for Biology of Aging in Cologne, Germany. And we are going to talk about a longevity drug that needs no introduction, rapamycin. Rapamycin, of course, has been shown to extend lifespan in various model organisms, and human trials are currently in the pipeline. However, there is still a lot we don't know about this drug. Scientists such as Xuan devise ingenious experiments to elucidate rapamycin's mechanism of action, and exciting new findings keep coming. So thank you, Xuan, for joining us. Rapamycin is considered one of the most promising anti-aging drugs, of course, despite some toxicity issues, and also the fact that we still don't know all the intricacies of its action. As one of the world's foremost experts on rapamycin, could you tell us why you believe in this drug?
So rapamycin, I would say, is one of the few drugs consistently showed lifespan extensions in diverse laboratory animals. We will talk about like the worm, C. elegans, fruit flies, and mice, and also other animals. And this is very powerful, especially those data actually from different independent labs around the world. So if you look into the very early paper, for instance, from Richard Miller and also some other labs from the C. elegans and also from our lab. So basically, Linda Partridge, we published a paper in Cell Metabolism in 2010. So we basically, actually this lab, because from different place, we actually saw that the rapamycin can effectively extend the lifespan in those animals. And I think more importantly, if I would say, because if we talk about the implication to human, is rapamycin has shown against some of the age pathologies, including cancers. So, and the second thing I would like to say why I believe in this drug is it is a licensed drug. So that means the base reactions and also the toxicity and also the ability and also the target, a single pathway is known. So that means that when we actually trying to propose these drugs for dual protections, it's kind of like a shortcut to help us to reach our goal. So those things, I believe that why the rapamycin is very powerful for anti-aging usage in the future, let's say.
Thanks for the great answer. And let's move to your recent paper that just got accepted to Nature Aging. It is mostly dedicated to the effect that you call rapamycin memory, when a transient rapamycin treatment in midlife recapitulates all the effects of a lifelong treatment, including lifespan extension. That's in fruit flies, but also some effects in mice. So please tell us more about this intriguing phenomenon.
Yeah, thanks. I mean, first of all, I would like to highlight that that is a yearly long work with an excellent team effort. So I was very lucky to be involved in this project when I joined the lab from Linda Partridge. And so namely that we have found rapamycin treatment of only 2 weeks in flies and only 3 months in mice in the early adulthood, extended lifespan in flies. And also we observed that reduced some age-related gut pathologies in both flies and mice. The important things that I would like to say is this treatment will get the benefit to the same degree as lifelong rapamycin administrations. So, and when we look into the detail, because obviously we would like to know a bit about the mechanism, we found that the autophagy, which we will always say this is one of the underlying mechanism, hopefully, under rapamycin treatment. And autophagy, if I would say, is a process that recycles or secretes cellular components, which is increased by the rapamycin. And very strikingly, what we found is even the rapamycin is withdrawn, the autophagy level still remained high. So how precisely autophagy levels stay elevated, like involved in we actually still don't really understand. But we found there's one little studied proteins known as alpha lysosomal monosidase. So we also call LMN5 in flies or MAN2B1 in mice. That is something, this protein seems to play a key role in this rapamycin memory effect in flies and also in mice. I think that the fascinating phenomenon, I would say, and also because the why was fascinating, we believe that because there's actually very important implications probably for the future, is we have found a way to circumvent the need for chronic long-term rapamycin treatment. And basically, it means like you have less potential negative side effects that might caused by the long-term treatment. But also, I believe that it's also it's also easy to prove to work in humans because it will be more feasible to be applied as we don't need to do a lifelong commitment and also investment.
Right. This is a really interesting observation because we know how hard it is to organize a human clinical trial, and the shorter it is, probably the better. Yeah, and we also, I think we should mention that fruit fly's lifespan is about 60 days, I think.
Yeah, that's more or less a 60
Yeah,
to 90 days.
Just the viewers know that 15 or 30 days treatment is not very short term. It's more like a quarter lifespan, but sure, that's fine. So according to a paper, it's another interesting finding that I found really fascinating. Elevation of autophagy by other means can also induce autophagy memory, right, and recapitulate rapamycin's effects. How would you interpret these results? And is it possible that studying the effects of rapamycin will actually help us find even better drugs that work along the same pathways?
Yeah, so actually that's a very good question, Vance. Just in my opinion, that is obviously fascinating finding that we found the short-term autophagy increasement can, maintain itself active in long run. So that is very funny and also fascinating. But to be honest, we still don't know much about that. So as I said before, we found short-term rapamycin treatment induced long-term autophagy activation in the model we used. But we also found that the short-term autophagy activations, or more precisely in our case, induce ATG1, so a protein that is able to initiate the autophagy process and directly controlled it by the mTOR. So that is something important to just keep in mind. And when we induce this protein called ATG1, it will keep the autophagy system goes on, and in flies. So that's what we observe. And from importance here, we would say that we observe this activation is sufficient to extend the lifespan and also maintain the gut integrity in the older flies. But as you mentioned, that I need to be fair because our research, we need to take the responsibility. So at the moment, we still don't know if this autophagy memory will achieve the same beneficial effect as a short-term rapamycin treatment. And we also have no data to show if autophagy memory exists in mammal as well, because I haven't tested in mice, for instance. So there's actually still a long way to go before we can say, yeah, now we can take this new approach to combat human aging.
Let's move to another very interesting topic, which is the sex differences in response to many life-prolonging interventions. So that's something that you report as well. We know that rapamycin works better in females than in males, although I have to say in mice, the difference is way less drastic than in fruit flies, right? Where you basically report no effect in males. So what are some possible reasons, in your opinion, for these sex differences, and what are the possible implications for longevity medicine?
Okay, that's a very good point. And first of all, when we talk about the sex differences, we need to go back to just look into the lifespan expectancy. You see women's always longer than men's in all the country. So that is also quite consistent. From the beginning of the record, 19th century or whatever. So if you go through the line, something you just found that there's probably not because the health, because you see that this benefit for women not only exists in United States and Europe, but also in relative underdeveloped countries like Africa. So that is something you need to keep in mind. But funny enough is that, or very important, this kind of longer lifespan in women seems to have a kind of like a trade-off, which means that the women suffer more chronic disease than men. So I mean, there's a lot of hypotheses. I mean, in the field, just in terms of the sex differences in lifespan or longevity, let's say. So it seems like the women can stay longer, but they suffer more because they can try to fight with, but just keep the body goes on. And in particular, when we talk about the possibility reason why we have this sex difference in rapamycin, for instance, and I think that is something I need to be fair. This is actually it's not only rapamycin have this sex difference, because when you look into the IDP program, for instance, this is kind of like a gold standard for assess the longevity drugs by using the heterogeneous mice to run the lifespans in 3 different locations in the United States, which is kind of like supported by the NIA. So they actually found that not only rapamycin, but also some of the other drug, like if I would say the acarbose, which is males have a more beneficial than females, and also the other couple of things. And when you talk about rapamycin, or we can also say more broad that not only rapamycin, but if you influence genetically, like let's say the S6 kinase, we also see the female extend the lifespan, but the male doesn't have almost nothing effect. So it seems like this sex different responses to drugs or to genetic manipulation or even to nutrition is somehow linked to some specific signal pathway in our bodies. So am I guessing, based on our finding, also the other labs finding, am I guessing is the difference is probably due to either the disease's susceptibility or the just differential importance of a signal path in aging.
Yeah, just want to point out that in the ITP, in the intervention testing program, actually rapamycin was one of the molecules that had the least sex difference. That actually, I think in lifelong treatment, the difference was really small. And in late life treatment, the difference was about 50%. But it actually, unlike many other molecules, it worked in both sexes. So I think that actually makes rapamycin more interesting drug.
Yeah, absolutely. And actually, that's at least I would say that even despite that in mice, so that's something we just talk about in mice. Maybe in human, that's a completely different story. But at least in mice, rapamycin work in both sexes. Just in female, they are basically stronger than males. But if you look into some other paper like the short-term treatment, for instance, from Max Planck, because they did this very late life treatment, you found that in this kind of treatment, the male gets better benefits than female. So there's always some kind of like interesting or maybe the interaction between the treatment and the sex. That means maybe, I mean, there's several explanation for that. For instance, maybe some of the signal actually changing with life. So that's obviously possible that because they always hypothesis like mTOR somehow get activated through during aging. And then when you treat them, start the rapamycin treatment at a young age or mid-age or even late age, you might hit slightly different targets. And the second thing that we talk about in flies, because I mean, I would say the fly is actually one of the best model because you can just, manipulate the sex in one or the other tissues. So the point is that because what we found in this case is the gut cell is quite important in response to rapamycin. So that actually brings up a hypothesis that maybe because during aging, I mean, obviously the organ, different organ, the aging rate is different. So that is actually, there's no doubt. So that means maybe when you start the rapamycin treatment and from different age, you might actually hitting, something different when you reach to the same organ. So you talk about the gut or you talk about the bone, but the young gut, the old gut is actually not exactly the same. Yeah. So that's something I would like to just say to keep in mind.
Yeah, and it was also very interesting to read about the effects of rapamycin in Drosophila flies. But their physiology, let's be honest, including their sexual dimorphism and also leading causes of aging and death, differs considerably from that of mammals. And this begs the question, how relevant studies in model organisms, and specifically in flies, are for us humans.
Yeah, so excellent points. I think that is always important to justify how important, how advantage of a fly and what's the limitation of using fly when we talk about the application, right? And first of all, I think there's no argument that the fundamental biological functions between fly and human are quite similar. So actually, if you look into the data, so around 75% of the genes responsible for human diseases are evolutionarily conserved in fly. So that means that they're actually fly and human, there's a lot, kind of like a big overlap in the basic biological functions. And it's also want to say that there's quite a lot of human diseases and nowadays can just use a humanized flies or just a genetic manipulation flies can just try to do the first drug screening or first stage screening. So that is something I just need to say. And the second, I would say that when we talk about the aging, right, we know that the fly also show the age-related decline, functional and structural decline, which is similar to humans. So we also have this mobility and also the memory and even the gut change is kind of like a decline with age. So somehow the similar age, maybe the speed is different compared to human, but still in general is more or less similar to humans. And I think there's also another thing I want to point out, and the fly actually has a similar response to DR, insulin, and mTOR signal pathway, and also the other well-known aging pathway, which is we compared to mice because we don't know what exactly human it is. But at least when you look into flies, or maybe even like a C. elegans worms and mice, you always found a lot of basic conservation responses, more or less to the same directions. And so that's actually one thing I would like to say why we use flies. And actually another aspect I would just try to mention, because there's always a debate or discussion. If you can use fly, why don't you can even use like a worm, because the worm actually live shorter, right? Compared to fly, one only live 25 days or more or less like that. So obviously there's a huge advantage. And when you look into the field, there's a lot of excellent researchers use worm to do the aging research. But just go back to the sex dimorphism. I think fly has unique advantages compared to the other small animals because it has the male-female sexual system. So XY chromosomes, similar to the mammals. So I think that is actually something just natural for flies. But obviously, I talk about a lot of advantage, but I have to say that it always happens some limitations. So those kind of like a disease development process between flies and human, basically, obviously not the same because they don't have some organs and etc. So basically, in my opinion, I think it's huge advantage to incorporate fly in this aging research or even like a drugs target discovery, and also do the high throughput and at the very early stage together maybe with the cell lines, because the fly is relatively easy to grow and also very cheap in my opinion. So there's a kind of like you can just save a lot of time and also money when you do the drug screening at the first sign. Because when we talk about the longevity, that's one thing I really think is very important why the fly or maybe C. elegans is the good model is basically because the aging itself is very heterogeneous. So doesn't like some specific disease, the aging, you know that when you look into the old people, just for instance, my grandmother, she's 90 years old, but she still can dance, can sing, and even can, actually she uses far more better when she, play the video game than me. So it's crazy. But if you look into some people, 90 is already very, some people, if they can, live up to 90, most of them may be already sick or have some diseases. So I'm saying is that the aging is very heterogeneous. And if you really want to study aging, you need to have enough statistical power to, mimic this kind of like populations. So to be fair, fly or maybe in also use a worm, this is kind of like this model you can create an army, let's say the army
The
for
army of
research.
flies. Yeah, that sounds good.
Army of flies, because you have like 1,000 and flies, and then you can just try to dig out how this heterogeneity is actually working for aging. And usually you can't, I don't think there's only number of a company or an institute can afford this if we're going to do mice work, because it's just crazy amount of cost and work. But obviously at the end, all these compounds after the screening from flies has to be, of course, validated in the the rodent model before we actually can bring it to the clinical trials. Yeah.
Yeah, that's a really interesting angle. And of course, aging is a very complex phenomenon. So we do need that big data and that predictive power. And here, yeah, flies fit in nicely, I guess. Okay, so in another paper, you demonstrate that rapamycin improves genomic stability via increasing histone production. So can we hear more about this, please?
Yeah, thanks. That's actually, I remember the lifespan. I actually reported this paper last year, so it's my glad to just share this a bit. So in that paper, actually, that's kind of like in line with the other two paper I just mentioned previously. In that paper, we use fly, basically that's our favorite model, and then we found that the rapamycin actually can increase the histone production or histone protein in the fly gut, which is somehow at the end change the chromatin structure and probably also the DNA, the way DNA storage and in nucleus. And all these changes at the end lead to there's some changes, histone marker changes and autophagy genes expression change. And this is kind of like this link. So what we found is called the mTOR histone autophagy link, which is we believe is unique because there's no one reported that before. And this link is kind of like a circumvent the classical ATG1. So what we just talked about this before, and ATG1 classic, the memory path. And this histone link somehow can also improve the lifespan and also the health span in flies. And more importantly, actually, we also, did some mouse work, and we found that in the mouse, we more or less see the similar phenotype. So rapamycin also increased the histone protein level in the mouse gut and also changed this autophagy gene expression, which we believe that at the end, we also try to help the health span. The funny things that I would like to just mention that because when we actually when I'm working on these cell papers, actually we got some requests to ask, so what's the difference between this histone signal pathway and the memory one, because that is ATG1. And we've actually tested whether this histone X can have a memory effect, because we believe maybe the histone methylation or this epigenetic things is something related to that. And unfortunately, at least in our trial, we don't see the histone have any memory effect. And so we propose that there might be rapamycin might have several different actions, like go through the histone or go through the classical ATG1 and then lead to some memory effect. But also that's something I would like to say, that's just we're looking at in the gut. So eventually, I think rapamycin even does more than, because rapamycin also reduce the cancer in the other organs, right? So that's definitely not only work on gut, but also some big organ. So I think there's still a long way to go. Despite that, the rapamycin in the field, we already have studied several decades, but there's still a lot of things can be found. But nevertheless, I think we are getting closer to use rapamycin or the other promising drugs for anti-aging usage.
And that brings me actually to my next question. As we said, aging is, of course, an extremely complex phenomenon. And many gerontologists think that, there's no magic pill. And the future of longevity medicine is basically in combination treatments. Do you have thoughts on possible combinations that might include rapamycin?
Yeah, absolutely. I think that's probably one of the best approach we have been tackling on. And so first of all, if we just go back to the classical like a polypill or cocktail treatment, we all know that it works in a lot of cases like HIV and also some cancer diseases. So there's no doubt if we say the aging is a disease or the mother of diseases, let's say, we definitely think that this polypill or cocktail will work to slow down aging. So if we actually our lab actually 2 or 3 years ago we published a paper that we combined rapamycin with another 2 drugs. So one is trimethylene, the anti-aging drug, and lithium. So actually, when we combine this drug, they're hitting different target of the insulin mTOR complex, let's say. So when you're hitting several different targets, actually you get this addition lifespan extension in flies. So that's actually amazing. And there's also some labs that using one, they also test the other compound. They also see this synergy effect of the drug combinations. So I think this is definitely the way to go. And obviously we can even have more, the creative idea that not only talking about the drug combination, but we can also combine drugs or pharmacological intervention with the other, like this senolytic or even like a cell therapy, because nowadays actually there's several different ways we believe that can extend the lifespan, hopefully in humans, but at least at the moment we can actually use this animal model to prove that. But on the other hand, there's always the challenging, because when you talk about the combination, you have a more and more possibility. So obviously that is not easy if we would just start this testing in mice, because it's just too expensive that no one can afford that, or just too long for that. So let's go back to my initial discussion, saying that we can actually use fly, worms, or some long, short-lived vertebrate like killifish, for instance, because they just only live several months. So we can actually do some high throughput screening or drug combinations and use this animal to test if there's a possibility to get some lifespan extension and also reduce age-related disease or pathologies. And then we can hopefully find some promising target and then move to the mice in the future to the clinical trials.
Yeah, thank you. That's another great argument in favor of flies, I'd say. So my last question would be, would you place your bets, let's say, on rapamycin becoming the first approved longevity drugs, and maybe you have an idea about the timeline?
Yeah, that's cool. I think there's a lot of betting whether rapamycin or other drug, for instance, like metformin, because I think that's also a very good example, like a TAM trial or pushing by the new Basilite, because actually I also talked to him several times. So I think that's also kind of like a very promising drugs, for instance. So yeah, I would definitely bet rapamycin becoming the first group approved longevity drugs. Whether that will be the first, this is hard to say. But I do know that there's several clinical trials for rapamycin is ongoing, like this pill trial, and there are also some other trials actually ongoing. So at the end of the day, whether that's become the first approved longevity drug, for me, personally, I think obviously I'm working on rapamycin, I will be happy to see. But I think for the whole population, for the aging population, that's not very important. The important is whether we can get the first approved longevity drugs as soon as possible. So we can, the elderly can actually get the benefit for that. So no matter whether rapamycin or metformin or even some other promising drugs, we just need to have it as soon as possible so that the people actually can get the benefit now, sooner than later.
Of course, I couldn't agree more. And thank you so much, Xuan.